Just to let you know that the seminar will be recorded, so please keep that in mind. And if you could also kind of stay on mute for the recording, that would be great. We'll have plenty of time for questions afterwards. I'd first like to acknowledge that the Statewide Biobank is located on the lands of the Gadigal people of the Eora Nation, and I'd like to pay my respects to elders past, present and emerging, and acknowledge the very important role that Indigenous people play in the lives of everybody in Australia and our society. So it's a great pleasure today to introduce Dr. Hugh French, who is coming to us from the Institute of Precision Medicine and Bioinformatics, just across the road from the Statewide Biobank within the Royal Prince Alfred Hospital as part of the Sydney local area health district. So I've been chatting to Hugh about his background. He has very interesting research background. He started his life as his professional life, I guess, as a scientist. He did his PhD at the ANU in the John Curtin School of Medicine, and he studied inducible gene expression in T cells, where he first became, I guess, exposed to methods that required bioinformatics, and so he finished his PhD as a sort of a bioinformatician, computational biologist. And then he did a postdoc with Professor Sue Clark at the Garvan Institute, worked on transcriptomics, and then undertook medical studies at the University of Sydney. And he's now, he's a genetic pathology registrar and working with Professor Ron Trent and Dr. Anthony Chung in the Department of Medical Genomics. So Hugh, I think, as somebody who obviously has, a fantastic background, really relevant in terms of thinking about how do we actually implement sequencing into the clinic, and how do we obtain the kind of resources that we're going to increasingly need in terms of data storage. So Hugh, I'm going to hand over to you. We're really looking forward to hearing what you have to say, and thank you very much again for talking to us about cloud-based analysis of long-read sequencing in thalassemia.
Thank you very much for the kind introduction, and thank you very much for the opportunity to speak to you today in this series. Today I'm going to talk about some of the main issues that we've had in trying to translate research tests, which long-read sequencing are, with a focus on thalassemia and how we're trying to develop cloud-based analysis solutions that we can implement in a diagnostic laboratory. But first, I'd like to acknowledge the traditional custodians of the lands that we're meeting on today, or I'm presenting from. And I pay my respects to elders past, present, and emerging, and recognise their ongoing contributions and connections to the lands and waters of New South Wales. I also acknowledge and pay my respects to our Aboriginal and Torres Strait Islander people and colleagues who are joining us today. And so what is driving the adoption of long-read sequencing in diagnostics is really a change in technology, which has been driven by industry. And at the Institute of Precision Medicine and Bioinformatics, one of our goals is to take these new technologies and translate them into clinical applications. Today, I'm going to talk about some research work that we're doing in the Department of Medical Genomics under the Institute of Precision Medicine and Bioinformatics to explore potential clinical applications of long-read sequencing. And this is done in collaboration with people we work with at the Garvan Institute, at Microsoft, and the Department of Innovation and Health here in the local health district. And I should mention this is partially funded by an MRFF headed by Ira Devison of the Garvan Institute in Darlinghurst, investigating the translation of nanopore-based testing into clinical diagnostics. Today, I'm going to focus on the analysis and informatic challenges of doing this, which is the focus of my involvement, using thalassemia as a case study. And I should mention that some of these slides are Ira's and some are taken from Nanopore themselves. So just a bit of background of diagnostic, workflows in sequencing. This slide is not Ira's, it's my own. Apologies for the poor photography. This is a photograph of our sequencing room in the laboratory here at RPA. And on your right, you can see a genome analyzer 3500 XL, which is a great piece of kit. that does Sanger sequencing, which has existed in various iterations since the 1970s. And this is first generation sequencing. On the left, you can see a number of sequences from Illumina, which perform second generation or next generation or short read sequencing by synthesis, which sort of became the dominant technology in the 2000s. And what's not in this picture, and we don't have, is a long read sequencer or third generation sequencer. So what I mean when I talk about long read sequencing or third generation sequencing is these three technologies, which are the ones that I'm aware of being currently available. Oxford Nanopore is one. Illumina has a solution called Illumina Complete Long Read technology, and there's the PacBio Revio. Today, I'm going to talk about Oxford Nanopore. I'm an AI on a grant, as I mentioned, an MRFF that utilizes Oxford Nanopore specifically. However, I have no financial relevant disclosures. My only income is the New South Wales State Award for a Registrar, which is perhaps not as much as you might think, but I certainly don't have shares in any of the companies or financial interests in anything that I'm going to present today. But I will talk about Oxford Nanopore. They're a company that makes one technology, and that's their nanopore sequencing. So we are trying to analyze, very large data sets when we're doing second and third generation sequencing, as many people who undertake this would be familiar with. When you have these big genomic data sets, one of our challenges at the Institute of Precision Medicine and Bioinformatics is that we have to do this analysis increasingly in cloud-based environments for both the analysis and the storage of data. This has led to the uptake of cloud computing locally, as it has across most of New South Wales Health. And sort of when you're in the eHealth multi-cloud, you have two real options in New South Wales, which is Microsoft Azure or Amazon's Web Services. To the uninitiated, which I was when I started this, one is Coca-Cola, the other's Pepsi. At least that was my understanding, and sort of they do the same thing. And everyone sort of uses the cloud when they come to work without perhaps noticing it. And that's true for us too in our department, where we already use cloud computing in commercial settings through New South Wales Health Pathology, where Illumina Base Space is a sort of complete solution for analysis, secondary and tertiary analysis of next generation sequencing data. We use products like Sophia and Answer Analysis. What I'm going to talk to you about today is when we try to take nanopore sequence data, for example, for the testing we're trying to do in thalassemia, there's no off-the-shelf commercial sequencing service that exists for these new technologies. So we have to make it ourselves, but implement it in a way that it can be run in the cloud, like our other solutions, and in a way that has the potential to be accredited in the future. So our story with cloud-based genomic analysis and third-generation sequencing started with a patient who we were unable to solve with current diagnostic techniques. The solution turned out to be third-generation nanopore sequencing. So then we were presented with a problem. How can we reproduce this? How can we take this custom bioinformatic workflow and implement it? And so the design, which I'll talk about, involves Nextflow using Azure Batch, which is a on-demand compute part of Azure, which is Microsoft's name for their cloud. And how can we do this in a secure way within the eHealth multi-cloud? And so this is an approach that we sort of take based on policy in New South Wales Health, where things have to be in a cloud-first environment. So what that means is we're not supposed to have sort of servers whirring away within the hospital. We're trying to increasingly migrate analyses into cloud-based environments. And so that's where our collaboration with the local team here in the SLHD who are working with us to enable precision medicine and genomic testing in the cloud, and take these research workflows and hopefully apply them in the diagnostic laboratory or the clinic. We were able to start doing this because our local team put us in touch with Microsoft's Fast Track for Azure team. So if you recall, Azure is the name of Microsoft's cloud offering, which is available within the eHealth multi-cloud. Then Fast Track for Azure is a program where if you're doing something a little bit unusual, like what we're trying to do with our genomic analyses, they give you tailored guidance from a special team of engineers who are sort of like the special forces, is how I think of them, of Microsoft's cloud architects. And they sort of gave us a design that allowed us to analyze our data, and I'll go through that in a bit of detail. But first, why are we doing this, which is our patient? So this gentleman had a history of beta thalassemia, was known, but he didn't have a severe thalassemia, so he wasn't transfusion dependent. And he presented with back pain and difficulty walking, and it turned out that he had a kind of severe thalassemia. So thalassemia is where you have either not enough or a non-functional protein product in your globin proteins. Your globin proteins, four of which make up hemoglobin, which is what moves oxygen around your body. You're supposed to have 2 alpha and 2 beta globins to do this optimally, and thalassemia can be caused by the deletion or variants affecting the production of these subunits. And genetic testing is how we determine how many functional copies of each of these proteins you have. So this is the spectrum of thalassemia disorders, going on the left from very mild, where you can be walking around, you're missing 1 copy and you're fine or nearly fine, all the way to requiring transfusion or even death. So it's a very broad spectrum, and just to sort of relate that this gentleman was looking initially like he had a less severe thalassemia, and as he progressed, he's now requiring transfusions, which didn't make sense with the genetic results we initially had. And it turned out that he actually had extramedullary hematopoiesis. So he was trying desperately to make more blood cells in places in the body where you ordinarily don't. In his case, around his spine, which was causing compression of his spine and giving him this presentation of difficulty walking and the back pain. And this wasn't reflected in his hematology or the testing that had been done to date. So he had a little bit of an anemia, but his blood indices didn't really make sense with what we were seeing in the genetic results. So he had a high fetal hemoglobin, fairly low HbA2, no abnormal bands, and he had only had identified a single deletion in the alpha locus, and what looked like on MLPA, which is a standard copy number test for the number of genes that you have in the beta globin genes, showed what looked like a large heterozygous deletion, but we weren't able to find out the extent of that using that technology. We didn't find anything using a traditional Sanger sequencing technology in the beta region. And so these results that we were getting from the genetic testing didn't make sense with the severity of the presentation that this gentleman had. So this is the MLPA results here. I won't go into how to analyze this data, but essentially, hopefully you can see my pointer. You would ordinarily, if you had 2 copies of beta thalassemia, have results along this blue line. Now, this gentleman had, where these red dots are, it looked like he was missing 1 copy of beta thalassemia, but he was sort of presenting as if he had 2 copies missing. And we didn't know why until we had a look at the nanopore sequencing data, which allowed us to find a deletion, inversion, deletion. So the skinny bars at the top there on one allele was a deletion with a region of an inversion, and then another deletion spanning from hemoglobin beta to hemoglobin gamma. And then in trans, he on the other allele had another deletion in beta globin. So this sort of now made sense. We couldn't see this exact region just because of the design of the MLPA, and we didn't have a probe that showed us that there were two copies at that exact location. So what I'm showing you here is this nanopore long read data, and you have separated it into the two alleles, two copies, red and blue. And you can see this region here, where the deletions overlap, that there are no reads at all. And you can see that this is concordant with the deletion up here. This is the big inversion. And then you have this deletion here. And this explained the MLPA results. If you look at where they're located, you can see that these two probes map to this part of the deletion, these map to that part of the deletion, and this maps to that. So we just never saw that two copies were missing using our existing technology. And so we're really excited. I should also mention that you can't see inversions like this on MLPA. They appear normal. Is a limitation of the excuse me. So this was a rare thalassemia that we weren't looking for. It had been reported in the literature a long time before, where one of my professors was a PhD student, Ron Trent, in the Wetherell Institute at Oxford. And so we were really excited and thought, it would be really great to detect these again if we needed to and offer it as a diagnostic test. Which led us to look into nanopore sequencing a bit more seriously, where these long reads allow us to detect these large structural variants. So that inversion that you saw before is something that nanopore sequencing and these long reads can map all the way across, and we get unique reads that span the whole region. And we can see the inversion and the deletions if they're around it there, which would be very difficult find using short read sequencing, and certainly we were unable to using MLPA, which is our standard diagnostic test. So the thing that makes globin regions challenging are that they, for example, in the alpha regions, have high homology. So they're particularly hard to challenge with short read sequencing because you're not sure where the reads will map within that locus. So very briefly, I've got a couple of slides that sort of say the same thing. Nanopore sequencing is where you drag the entire DNA molecule through this pore. And as the DNA passes through that pore, the different bases cause a change in current across that membrane, which is essentially measured on every pore on the flow cell. And that current is turned into what I call squiggles, these squiggly lines or changes in voltage, which are deconvoluted into base calls. So essentially, as the DNA whizzes through the motor protein and the pore, the change in voltage is measured and this is turned into base calls. What this means is that your read can be as long as the DNA molecule that goes through the pore, which is why it's a long read technology. And that's just sort of another sort of visualization of it there that just shows that these flow cells are actually made up of many, many of these pores. You can have one pore active in a channel at a time. The small sort of USB size that you might have seen around is 126 channels. A MinION, which is a bit bigger, is 512, and a PromethION, which is a big sort of 24 at a time or 48 at a time sequencer, has more channels. And so just to reiterate, you take the entire length of the molecule that you can get through the pore. This creates voltage changes, which are turned into base calls, which is what we work with. And just to give you an idea, if you've not seen nanopore sequencing before, this is from Nanopore themselves, so it's what they sort of claim. If you're just doing, for example, a standard whole genome sequencing, which we did initially, you get around 30 to 40 times coverage using one flow cell. And then there's sort of different applications you can take from there, all the way up to very high accuracy duplex cells, which I'll touch on later. Or if you want to get even higher coverage, say you want to do somatic testing or something, then you start looking at more than 2 flow cells a genome, and sort of other applications can have less flow cells and more genomes, which I will talk about in a minute. One of the issues with nanopore sequencing that sort of delayed its adoption in some laboratories was the accuracy, which, this is again nanopore data, so you have to take it with a grain of salt, which they now say is very high for SNVs. And when you're using the high accuracy base calling with the simplex, so just one strand going through, which is what I've presented, the indel calling, is improving, but not quite at the same level as you might see in Illumina. So that's for small structural variants, but structural variants detection is actually quite good. You don't necessarily want to do a whole genome though, if you're just looking at thalassemia. And so this is a technology that Nanopore has created where the DNA molecule is dragged through the pore, and the first 100 or 200 or so, I forget which, bases are read out. And if those bases don't map to your region of interest, then the current reverses and the DNA is spat back out and not interrogated any further until another strand, which is in your region of interest, is detected, and then that whole strand is sequenced. So you're not wasting your sequencing real estate on those regions that you're not interested in. And this is sort of when you can start just looking, say, at a small subset of genes without wasting all of your sequencing on other parts of the genome. And so this is just another depiction of that. You upload the areas that you're interested in. That's called read until or adaptive sampling. If it's not in your region of interest, then the strand is rejected or spat out of the pore. And then you wait for another molecule to come along. And if it is in region of interest, then you keep sequencing. So this allows a sort of enrichment process for your areas of interest. For example, in our case, the thalassemia region. So when we take this patient and don't do whole genome sequencing, you can see we still get acceptable coverage that allow us to detect the variant in the region. And so instead of using a whole Promethean big expensive flow cell, we're now using a smaller flow cell, and sort of our challenge is to get more and more samples onto one flow cell to start to make this cost a little bit less. This is an example of the very high homology, it's the wrong slide, HBA region. And this is the untargeted regions. So this is what the area looks like where you've just got these short little reads where they have not mapped to your region of interest. So they've just been spat out, and you end up with these little sort of fragments, and you're not wasting all of your time with that molecule being sequenced going through that pore. And so this data is now becoming more and more accurate, as I mentioned. And this is a slide from Ira Devison's group at the Garvan, which has just shown these dramatic improvements in the technology in terms of the SNV calls that you get from 2018 through to the contemporary super accuracy modes, where you can see that what the manufacturer is saying is true. There are dramatic improvements in accuracy. And this is sort of an example of the read lengths that you can get with a sort of not too onerous DNA preparation, where you're sort of getting, a lot of reads in this 50 to 200 kilobase length, which is not the longest that you can get, but is usually good enough for most applications. And for example, in some of the diseases that we're investigating, like thalassemia, you don't really need something longer than 15 to 20 kilobases. So it can be sort of a trade-off between how hard you want to try to get really long reads if you really need them that long, or if you just sort of need long reads that are fairly long, but not super long. And so this is just another depiction of the improvements in sequencing over time. And what I've shown you is simplex data. We don't have any experience with duplex data, but I will mention it briefly because it's supposed to lead to quite an impressive increase in accuracy, where you're actually sequencing both strands of DNA together and incorporating that into improved base calling. And how that works is your strand floats along with the motor protein. You sequence one strand, and then it floats back in, and you sequence the other strand. And so that's duplex sequencing. It promises to increase accuracy. We've not had an experience with that, so I won't talk about that further. What I will talk about is the data analysis. And so what you start off with is a POD5 file, now it's called. Used to be called a FAST5. Various other iterations, such as SLOW5, exist. And this essentially is a big file that contains all of the squiggles or the electrical current data from the nanopore that you turn into the base calls. And so this is the first step in the processing, which usually takes place on the machine or next to the machine. Which is quite a GPU intensive process. And so this is what generates the FAST-Q files, which those of you who've worked with next generation sequencing data before, FAST-Q files are the actual sets of reads or the base calls that you map to a reference genome. So FAST-Q is a file which has all of the bases and the quality of those bases, and this is the data that you use to map your reads against a genome, which is the first step of the secondary analysis that we perform in our cloud-based environment. So I've got a depiction of that here where you have these sets of reads, and this is sort of similar for short reads or long reads. This software Minimap 2 is actually something that you use for long reads. It's been optimized for that, where you map the reads against a reference genome and you find out where they match and mismatch. And then we use a program called Sniffles2, which gives us these structural variants, which runs in parallel with a program called Clear3, which detects SNVs indels, which also takes place in parallel. How that looks in an architecture diagram is you've got your FAST5 or POD5 file that is the raw data, which is converted into the FAST-Q file, which are the reads, which you then have to uniquely map to the genome, which gives you a BAM file. For those of you that are familiar with IGV or other visualization software, that's what you look at in the integrated genome viewer, where you see all those reads piled up, like I showed you on the diagrams before of the thalassemia region. And then you use that to call the different structural variants using Sniffles2, which gives you a variant core file, or the SNV indels are put into Clear3 and put into a VCF. And we also get phase data from that. And by that, I mean you can determine, because you've got long read sequencing with multiple SNPs on it, which read belongs to which allele, the one you got from your mum or the one you got from your dad, which is also useful information not always available in short read sequencing. So Nextflow is sort of becoming the lingua franca of orchestrating these bioinformatic pipelines in cloud-based environment, where it allows you to take the same code that can run on your laptop slowly or your desktop. You can run it in a high-performance compute facility, or you can put it in the cloud. So it allows you to take analyses that you've put together from one computing environment and run it in another. And this is what we would like to do with our bioinformatic workflows, where we may develop it, on a laptop or at a high-performance compute facility in the Garvan or the Sydney University, for example, and then, but also have that same code be able to run in a cloud environment. The way that we were able to do this was with the help of Fast Track for Azure, which is the Microsoft team that I mentioned before. One of the problems with bioinformatic workflows that we have in a research environment is that they are not necessarily secure because it's not something that's really been built into most research components. This code allows you to take the encryption and the passwords that you have to secure software in cloud environment, such as that we have in the eHealth cloud in New South Wales, and use it to secure your Nextflow pipelines. And so this is what we're working with Microsoft to do, who have also built a graphical user interface to allow us to interact with this process, requiring you to log in a secure way to run your data. How this is built is you take the bash commands, which are a series of the different workflows that I put before. So bash is what you type into a command terminal or a terminal on a computer, for those of you that are familiar with the command line. These are wrapped up into Nextflow, which is another language that takes those commands and puts them in order and allows you to run them in what are called containers, which is sort of like a virtual computer that just exists within the cloud that only contains the software that you need to run that particular process. So it's sort of a self-contained computer within the cloud that just has all the data you need and all the software you need to run your particular workflow. For example, the Minimap software or the Sniffles software or the Clear software that do the different Varia processes. How we build this is by having our repository with our code in it. We build the function that we're going to run, which is all of the code that we need to perform our analysis. And this is run in something called Azure Batch on the files that exist in our secure cloud, because this will be patient data we're dealing with. So within the health cloud where the patient data is stored that will come off our sequencer, it's then run on demand in Azure Batch, which just means, you use batched commands to just run one particular workflow on these containers, which are the virtual machines one after another. And I'll show you in detail what that sort of looks like. You have your secure Azure instance or resource group, it's called, which is your own little part of the eHealth cloud. You build the function, which is the contained workflow of mapping uniquely and calling SNVs and CNVs. You build those virtual computers and you put them sitting in the cloud ready for you to call whenever you want to run an analysis using Azure Batch. And that's done using Azure Nextflow, which just allows you to take your commands that you've put together in Nextflow and run them securely in Azure. And so this is the web interface that I mentioned before, which is how you actually interact with all of this. And that is the function here. So the lightning bolt that is the trigger for the Azure Batch jobs to act on your securely held data. And so this is what your directory looks like in your cloud, where you have your sequencing FASTQ files just sitting here, ready for you to run your commands on. And so then you have to log into your web portal, which you have to use your New South Wales Health multi-cloud credentials, because remember, this has to be a secure process. And this is what that looks like. So this is one of these Azure Nextflow user interfaces where you see the login button up there, where you log into your function. You get a splash screen that would be recognizable for those of you that are my colleagues in New South Wales Health. You log in using your privileged credentials, and you add the dispatcher, which is the function, and which pipeline you want to run or workflow. So this is all of the code that contains the instructions to run Minimap, sniffles, etc. And so you load all that up and you're ready to go. You put in the file name and the name of the sample that you want to run, and you send your job off. And so there's no resources that are sitting there waiting. This is all created on demand using Azure Batch, where you send the job off and it goes through to running the analysis on Azure Batch, and then you will have in your file storage, which is your directory in your secure server, the BAM, the VCF files containing your reads that have been aligned, your variant call files for SMVs and structural variants will appear, and they have not left your secure cloud environment. This is just to give you an idea of what the code looks like, an abstract term where I talk about it. And this is from James Ferguson's GitHub repository, which is publicly available. And you can sort of see, this is a parameters sort of definition file that's what's built by, these workflows, and allows you to take code that's generic to run on any sample, and you plug in what reads and what sample you want it to run on every time you do an experiment, and you're running the same analysis over and over again. And so this is just a generic description of what that looks like. What I showed you on the previous screen, these sort of arguments here where you have your parameters, which are what is this program going to act on, which in our case are the reads, and you give it a sample name, and it gives you all of your outputs, which appear in your secure directory. And this is just an example of what it looks like when it's running. It gives you a message back that says, we're running this command on your files, and you can expect your results in one of these folders presently, which depending on how big the reads you put in and the resources you allocate can really vary. So you can give the job a whole lot of cores and a whole lot of RAM, and it'll run very, very quickly, or not so much, and it'll run slowly. And an advantage is that because it's Azure Batch, you're not using one virtual machine, it's lots of independent different processes. You can actually run many samples in parallel if you'd like to. So where we are at the minute are that we've created a prototype with this full workflow. We've put some public data through so that we know it's worked, so publicly available nanopore sequencing data. We've added the secure authentication and data transfer and implemented it in our cloud, which I've shown you before. And by our cloud, I mean the Royal We, New South Wales Health eCloud. And so now we're in a process that's sort of at the limit of my expertise, or well beyond it, I should say, where their team is at the SLHD is looking carefully at this to make sure that it is something that is secure enough and meets their requirements, that we could, put some other samples through and see if it's something that's potentially an accreditable test. And if we get to that stage, then we could perform a validation, for example. But what's really exciting about this is that if we do manage to get an accredited test, we will have created a sort of a process where we can change the code that we put in at the beginning and run it in the same secure way, and potentially add further diagnostic tests in the future, and that other people might be able to do the same thing because this code is all generic and can be run in anyone who has access to Azure, which is anyone in the local health district and beyond within the New South Wales eCloud. So I'm a very small part of this work. It's led by Professor Ron Trent and Dr. Ira Devison particularly. The patient I showed you was one of Joy Hose and Stephanie Anderson, who've been involved in this work. The thalassemia team, Anthony Namali Stewart, various people from Ira's lab, Igor, Gillian, and James, particularly James Ferguson, to emphasize his work. He's the lead bioinformatician on this. And Ronel Veloso and Alex Gonzalez have been critical for Microsoft Fast Track for Azure. And we really couldn't have done anything without the local team here in IT and infrastructure, crowd infrastructure. Graham Stutton, Pauline Reddy, Artie, Steve, Drew, and Mitch Berger, who's been a great advocate. And these are the guys that actually have done all of the nanopore sequencing that we've used. This is the Devison group. Photo used partially with permission from their recent end of year celebration at a bowling club. And so this is the team. They're a very talented bunch, and they run a service sequencing they offer nanopore sequencing in a fee-for-service basis. If you ever have the need, please reach out to Ira and his team, and I'm happy to put people in touch with them. Thank you very much for your time. I hope that wasn't too computational, but it does give an insight into the challenges that we have when we try to take tests like this great research test we had for thalassemia, and we really want to offer it as a clinical service, and try to find a pathway to do that in a way that meets the justifiably stringent requirements. So thank you very much, Professor Byrne. I'm welcome to take any questions if there are any.
Thank you, Hugh. That was really interesting. I'm sure there'll be some questions. So if people would like to ask a question, oh, yep, Brent, I can see you've got your hand up. Please go ahead.
Hey, Hugh. That was a great talk. I actually did my
Thanks.
honors at Garvan with the love and support of some of the people you've worked with there, James and Hasindu and Gillian, I think in particular.
Oh, they're very, very talented bunch, particularly Hasindu and James.
Yeah.
The work they do is amazing.
Yeah, they're one of a kind. I was kind of curious. I know one of the big challenges with cloud workflows is obviously getting the data up into the cloud. My sense is that you were doing it only on FASTQ files. Are you putting any of the signal data in the cloud?
No, so there's two reasons. Firstly, for simplicity, we can use sort of your normal vCPUs for the mapping process and the structural variant calls, etc. Whereas the signal processing requires GPUs, which we could potentially put in the cloud if we had very low latency. Or it's just easier to run it on the machine. Now, there is a delay there where you run it in the cloud, and so it would make it difficult to implement the adaptive sampling or the read until if you didn't have the GPU right there, so you couldn't instantly read those first 100 base pairs and then feed back to the sequencer, spit it out. But if you're doing a one-way flow of information, then I think where people have done ultra rapid nanopore sequencing in international contexts, like you and Ashley's group at Stanford, where they just push it up one way, and they can deploy a huge number of GPUs in the cloud environment to process that data very quickly, which you'd want to do, particularly if you're using the super accuracy calling rather than the high accuracy calling. Then we'd start to look into the cloud, but we're sort of trying to build a minimum viable product here initially, and GPU at the moment is on premises next to the machine, particularly with adaptive sampling. Does that answer your question?
Yeah, that was fantastic. Thank you so much.
Oh, thanks.
I've got a couple of questions, Hugh. My first question is, and I'm just going to ask these questions while other people are thinking. So the first question, I guess, is about the nanopore technology. I know that a lot of sequencing platforms have issues with runs of the same base.
Yes,
and
so does
this. Yeah, I was going to say, I could see that
so
might
it's
be the
sort of like,
here.
the iron torrent has real trouble with it. Nanopore sequencing has, sorry, I did skip a bit when I said has trouble with indels. I should have said particularly homopolymers, where you're sort of seeing the same base come over and over again. It is getting better, but the computer does struggle to tell that same squiggle, how long is a run of A's.
Exactly. Yeah, no, I could have envisaged that.
As the algorithm becomes better and better, hopefully we'll see an improvement in that.
Yeah.
Fortunately, there's not many diagnostic applications for the size of those. One application that is sort of great interest in putting on nanopore is repeat disorders. And, they seem to be okay with doing repeats
Yes,
where the
as long as it's
base does
different
change.
bases.
Yeah.
Okay. Yeah, no, that's interesting. I guess the other question that I had was, I suppose in terms of a workflow in future, there's still many disorders that can be diagnosed without the sort of long read technology. So at the moment, it sounds like do you sort of envisage a situation where people will say, well, people will want to use long read, but they'll need to show that they have kind of exhausted all options before they progress to long read, or do you think long read will eventually just become what everybody uses?
So I sort of glossed over this. The reason that we've sort of introduced this as our first sort of test is that, we're the reference laboratory for thalassemia in New South Wales. This is our test of last resort almost at the moment, a research test, but, it's not going to be economic for us to do as a first line test for an MBS thalassemia genotyping study at the moment, but it is something that we like to have in our toolkit in those rare instances where our multiplex gap PCR, our MLPA, our Sanger sequencing have not yielded an answer, and we have a professor such as Joy Ho or her colleagues here at RPA who are still searching for an answer. We would like to be able to offer this in a diagnostic workflow as that final test that may not be economic for everyone. I mean, in the future, it would be wonderful if you could have one of those little USB size sticks, and that someone could be doing this on a laptop in, some malleable belt country and using the cloud to analyze their data. But that's sort of a long way from where we are now. But, it does start with a single step and a single test.
Yeah, definitely. We've got a couple of questions coming in, so I might ask a question from Umesh. How do you compare this with droplet PCR?
Digital droplet PCR, we have accredited in the laboratory for various tests, and we might be looking for an amplification or something, but sort of this is a test that we apply to, these either whole genomes or a large panel or something like thalassemia, whereas digital droplet we're sort of trying to interrogate exactly how much of something is there. So the coverage that we get for nanopore sequencing is too expensive to sort of do somatic testing, for example, where you might want to use digital droplet PCR to really find something a low variant frequency. We're not going to do that with nanopore. It's too expensive. Was that sort of what you were angling at, Hugh?
I think that was Umesh. Yeah.
Oh, sorry. Apologies, you mentioned.
Yeah, no, that's okay.
Great.
Yes, looks good. Yes, thank you. Okay, Hugh, please, yeah, go ahead.
Hi, Hugh. Great talk. I was just wondering, do you think the nanopore sequencing will ever reach the same sort of low error read rate that Sanger sequencing has currently?
So it really depends on the coverage. The more coverage you get, because the errors are sort of random, you can get up to very, very high quality scores and very error-free sequencing. Will it ever be better than Sanger sequencing, which is sort of the gold standard? I think once you get up to very high levels of coverage and very high quality scores, it sort of becomes academic. I think that it's really a question of coverage. You can do a lot of coverage, like you can in the PacBio Revio system, where you get very, very high depth because you're sequencing the same thing over and over again. Or if you really sequence something to death on nanopore, then you can get up to those very high quality scores. Will it be able to get very high quality sequencing that's as cheap as easy as Sanger? I'm not sure. Sanger has still got a role in diagnostic laboratory where you really want to see if a variant's present or not.
Yeah,
But you get very high
yeah.
qualities now with nanopore sequencing. The SNV data is looking increasingly better and better and better, and the duplex data the company promises will be even better. So Maybe.
Does anyone else have any questions? We've still got some time. I was going to ask a question about something that you said right at the beginning, which was about I remember you said there's changes in technology driven by industry, which I thought,
Yes.
I mean, of course, technology advances are probably going to come from industry in this space, but I'm just wondering to what extent, it could sound a little bit like tail wagging the dog in a way, like to what extent are sort of human applications driving these technological changes?
So
Or
I
the
think
human needs,
I think we've been limited in what we can see in terms of human disease, genetic disease, by the technologies that we've had. So initially, we were only able to, do linkage and look at one gene at a time and things like that. And then now we've been able to look at whole genomes. We sort of don't know what we've been missing with those short-read genomes. And I imagine, but no one can be sure, that there might be a period of, disease discovery where we start finding structural variants and variants that we were simply missing because we were limited by the technology that we had. So I think that, yes, technology is driving it, but that's sort of how genetics has always been since the invention of the carrier type. Someone figured out how to do it, and then they discovered all of these diseases. And so, I think that there is some utility to that, but I accept what you're saying is we are sort of reactive to the change of technology rather than asking for something that doesn't necessarily exist and getting it built to solve a problem, perhaps because we don't know the extent of the one that we have.
And do you think that this is kind of where biobanking could come in? Do you think that industry can access the sorts of samples that it needs for these kinds of, unusual analyses?
Absolutely. So, look, I mean, biobanking, particularly, if you have the ability to freeze something, for example, instead of dunk it in FFPE, which is not a criticism. I understand that that's, an essential part of, anatomical pathology workflow, which is where we get all of our somatic samples. But, you want to sort of not do those sorts of things to samples that you might want to do long read sequencing on in the future. So that's where biobanking comes in, where you're doing something that's sort of a level of complexity more sophisticated or outside the clinical workflow that might be standard, which is like, get a sample, put it in an ETA tube and then freeze it and without doing anything to it. And all these sorts of things degrade that high molecular weight DNA that you really need to perform at the best quality long read sequencing. You may not need it for every application, but certainly that's where good biobanking, is critical to good data, because we're dependent on the samples that we get referred.
Thank you. I think that gives us something to think about because, here at the Statewide Biobank, we're storing samples for future research, and you never really know what future research is going to require. So, yeah, no, that's a great point. Look, I think
Well, I mean,
yeah,
in the future, even more challenging again, one of the exciting things about nanopore that I didn't get a chance to talk about today is that you can directly sequence RNA, which makes, how the sample's been extracted and stored, even more challenging. Again, don't want to beat up on FFPE tissue, but, getting good quality RNA is another challenge in addition to usual DNA extraction.
that's really, really great talk. Thank you very much, Hugh, for your time. I learned a lot. I now actually know what nanopore sequencing is. And I think that, the work that you're doing to create these workflows in the cloud, it's clearly complicated, but it's the sort of thing that we never see. And it's absolutely necessary if we're going to be able to, put these new capabilities within health and draw benefits from them. So, yeah, congratulations to yourself and the team, and it was lovely to hear about your work. Thanks to everyone
Thank you
who
very
came
much.
along. A pleasure.